Thanks, Olga. Thanks for having me. I'm very excited to be here. So I'll talk about sort of active touch, active sensing in elephants, and the angle of the presentation will be different from most of the other animal work that you may or may not know about. A lot of the animal work in neuroscience these days evolves in a relatively few model specimen, a small specimen, rats, mice, worms, and these model specimen, they are big success, what is unhealthy, however, is that all the work is done on this model specimen, or not all the work. I would say 90% of the people and 95% of the money, it goes into very few model organisms. And also, this model organism work, it's often not profoundly biological. It's not driven from the biology of the animals always, and it's unhealthy. And so today you will see something different, non-model organisms, so the classical model organism, elephants. I think elephants are great models for having a great brain, They have a big brain, and we really don't understand this brain, and that's what attracted me to them. They have also this fantastic touching behavior. And what we'll do is, what is good in the mice or in Drosophila, you can work on very particular questions and get very definite answers. It's very definite answers. Very hard in elephants. So it's more sort of guesswork today a bit. We guess. I think we have a hunch, but we ask broad things, broad questions, big things, because elephants are big things, so big questions, And what are the big questions? Sort of, what's the origin of intelligence, elephant intelligence? That's something we want to consider today. Or what do they have this big brain for? It's something really we do really profoundly not understand, and understanding why animals have big brains, it's interesting. It's obviously genuinely interesting. There'll be three parts. I talk about elephant species and grasping and the trunk. I will not cover fully our work on the trunk. I think I gave out papers on some of our muscle work. Unfortunately, we will not talk about. All of that, what we do here, we couldn't do without Thomas Hellebrand. We get the elephants from him. We get 3 to 4 elephants from him every year. Makes a big difference. Then I talk about elephant brain size and cellular structure, and finally tool use, house tool use in elephants. So to start, we start with a movie, so that we have this active touch thing. Some of you might have seen it already. It's happening there. And there is Pang Pa. She's in the zoo, a kilometer from here. She lives. And when you give her a yellow banana, she eats it. Just like every other elephant. That's Lena who did this work with her. When you give her a brown-yellow banana, however, she peels it, and it's 100% contingent on that it's brown-yellow, she peels in a very interesting way, What she does is she holds on to the peel and shakes out the pulp, So now the pulp is out, she collects it, and then she does a couple of checks, She does a lot of checks here. Now we look at it in slow motion, and you can really see, now she wants more if she brings up the trunk, we look at it in slow motion, you really see what's happening, She breaks first the banana. It took us several months to see that she breaks every banana. And the thing is, they do it so fast. When you look at it here in slow motion, the trunk, it reminds you a lot of the hand. It looks a lot like the hand. When you look at it in real life, it just looks fishy. You can't really see the trunk. And what's the reason? I think the reason is that they move their trunk so fast. For example, here Pa, she peels on average a banana about 3 times as fast as a human. And this is impressive, for the robotics people here. A human is not a bad robot for peeling a banana, So they're really, really fast, here I only want you to take these wrinkles. Yeah? I'll talk about these wrinkles quite a bit. Yeah? She does a couple of checks. Yeah? There is something, a fascinating backstory to this. So she came as an orphan elephant to Berlin, yeah, and why does she peel bananas? She's the only elephant in the zoo here that peels bananas. Also in other zoos, they don't peel bananas. On the internet, you can find banana peeling elephants, but it's rare, it's very rare. Why does she do it? She doesn't do it very good. We think her caretaker, she was this little princess, half-a-year-old baby elephant, and he always peeled the banana for her. And then a couple of years later, she came up with this algorithm to peel the bananas. Yeah? And it's sort of a cross-species behavior, very rare. I worked for 30 years with rats and never took up one of my behaviors. Another thing is they are very... the elephants, they're very different from one another, and I'll talk more about this. There are 3 extant species of elephants. Yeah? There used to be more elephant species, 5 or 6. They all disappeared when humans appeared, mammoths, mastodons. Probably the humans got rid of them. Also, these are endangered. There are quite a few African savanna elephants, yeah? The largest elephant, about 400,000 or so. Maybe 100,000 African forest elephants. I will not talk about these. Then there are Asian elephants. I'll talk quite a bit about African and Asian elephants. And in school, you already learned the Asians, they have the small ears, and the Africans have the big ears. Yeah? Guess what? I don't think this is the most important difference. Yeah? I'm going to give it away if I don't forget about it, what I think is the most important difference between these 2 species. Yeah? The elephants, they live in these tightly knit, socially very coherent groups, yeah? And they have very strong bonds, and I think they intensely compete with each other in these groups, yeah? And I think this might play into many things that we see today, actually also into the active touch, and I'll talk about this in a second. All elephants, very important. They have this long life expectancy, a low reproductive rate. The behavior is very experience dependent. Now, this is much different from the classic model systems in neuroscience. The classic model systems in neuroscience, mice, Drosophila, what have you not, they have all short generation times and a lot of offspring, and that makes them genetically tractable, yeah? But if you have a lot of offspring, and if you live very briefly, you can adapt to environmental change by genetic mechanisms. The elephants, they cannot do that, yeah? So I show you later a video of Mary. She's the oldest animal in the zoo, yeah? They live a long time, yeah? She's end of 50s, yeah? So they live a long time, so they need to do experience-dependent adjustments of behavior. And that makes them very interesting. So, elephant trunks. When you're a very large animal, you need feeding specialization. And the reason is that when you get bigger, the food, it doesn't grow bigger. Yeah? So you need to acquire a huge amount of food, about 150 kilograms or so for a large elephant. You cannot do that by conventional means. So all animals that are very large compared to their prey, they need feeding specializations. Like here, the blue whale, they have the baleen, or the sperm whale, it has this deep diving. For the elephants, it's the trunk. Yeah? They are very specialized on their trunk. And the trunk is a fusion organ from the nose and the upper lip. Yeah? This fusion, it happens in the 4th month of pregnancy. Yeah? This is a 3-month-old elephant fetus that just have a long nose. Yeah? The lip is not yet fused. Yeah? So first they have a long nose, and then the nose and the upper lip fuse. Yeah? There are interesting differences between the two, between Asian and African elephants in the way they grasp. And they relate to differences in the trunk morphology. So the African elephants, they have two triangular protrusions at the tip of the trunk, a dorsal and a ventral finger, yeah? And the way they use these two fingers, they call them trunk fingers, is they pinch things, yeah? They do pinching, huh? Now, the Asian elephants, they have only one finger. There's also a triangular protrusion here, a dorsal finger, but no ventral finger. It's more a bulb down here. And what the Asians then do, they also can pinch. You, for example, saw Pa pinch a little bit in a movie before. But when you challenge them with a big fruit, like a melon or a mango here, they would like to wrap their trunk around the fruit. Yeah, so they collect it differently. And these behaviors, they very much relate to brain structure and trunk structure in the two species. So they also relate to the wrinkles. So the Asians, as you saw also in the video, they have very many trunk wrinkles. Yeah? The Africans, they have noticeably fewer, yeah, of these wrinkles. And this is a paper that we did with a former colleague of our next speaker, with Andrew Schultz. In the distal part of the trunk, yeah, the difference is most pronounced. Yeah? Here, the Asian elephants, they have very many wrinkles here distally. And what we think is the function of the wrinkles, they make the trunk more flexible, yeah? We think. And so we think this is an adaptation to this wrapping strategy of the Asians, yeah, so that it's easier to wrap, yeah, whereas the Africans pinch. I talk even more about the wrinkles if I have time. Now, a lot of the neuroscience work, it totally unfolds in the lab. We at least have also some connections to the field, and the next result is a finding from the field that I want to show you. And it's very interesting. I think it's very dangerous and very problematic that there is so little field work in neuroscience. Yeah? There are thousands of people working on mice. Yeah? You would think there's any mouse field person that looks in the forest at the mice? Nobody. Yeah? And it's a big mistake. It's a big mistake for active sensing. Why? Look at this here. So this is from the zoo, yeah? An elephant from the zoo. And this is an elephant from the wild. Yeah? This is an elephant we anesthetized in the field. We put something in the trunk. You need to keep the trunk open. Elephants die when they can't breathe through the trunk. Yeah. So the first thing is when you anesthetize the animal, you put this stick in the trunk. If you ever anesthetize an elephant, put a stick into the trunk. Yeah. And now look here at these zoo animals. Yeah. Lots of whiskers, very long whiskers. Yeah. And all zoo elephants, African zoo elephants, Africans have these prominent whiskers. Yeah. They all look like this, yeah? And when we wrote a paper on this, and we had also a colleague who wrote a paper that maybe these whiskers increase the action space of the trunk, the sensing and stuff, yeah? But now, guess what? When you collect the animals from the field, the whiskers are all gone, yeah? And it's because the elephants in the field, it's a hard life, yeah? They sort of feed about 18 hours a day, 16 to 18 hours, and they're constantly feeding. Yeah? Most mammals for feeding, we sit down. Yeah? Also other mammals, at least they stand. Yeah? And they feed. Yeah? The elephants, they have this. They collect always as they go along. Yeah? Because you just can't come up with these 150 kilograms of food if you take time to stand. Yeah? So here it looks like treated with sandpaper. Yeah, the structure of the skin. There are no whiskers, first of all. Second, it looks like it's been treated with sandpaper. And guess what? every elephant from the field looks like this. Yeah, we collected a bunch of trunks from the field. They all look like this. All the zoo elephants look like this. So a giant difference. So it's very dangerous when you study touch or so. I just think this doesn't give you the full picture of how it really works. Yeah, usually the native state is probably this. Yeah. Okay, more about grasping. There is this left and right trunker behavior. Now, this is again trunks from zoo animals. Yeah, you see the long whiskers. An African elephant, Asian elephant, one finger in a bulb, two fingers. Yeah. What is very striking is vibrisal asymmetry. Yeah, here, look, the whiskers very short, very long. Very short, very long. It's not that we shaved it. Yeah, the elephant died. We get the trunk. Yeah, it looks like this. Yeah, no whiskers, long whiskers. Why? This is a reason. They have this left trunker and right trunker behaviors. So they are very lateralized behaviors. Yeah. So Anjali, she's sort of what they call. I always mix left and right up. I need to really look at it very carefully. I think she's a right trunker, so she would have very short whiskers here on the right side of the trunk. Now she grabs to the left, yeah, and then these whiskers, they are on the ground, they get abrased, yeah, and these stay long, yeah. That's how you get these asymmetries. And the elephants, incredibly lateralized behaviors, much like humans. There is an important difference, though. In humans, about 90% righties, yeah? In elephants, it's 50-50. It's random, not genetic, mother, child, random. Yeah, 50-50 left trunkers, right trunkers. This is something that makes the grasping behavior of the different elephants more different, yeah? So they have a very, very different way of grasping the individual elephants, yeah? Cool. This was sort of what I wanted to say about the grasping. I go now a bit into the brain, yeah? And we talk about brain size and the cellular structure, yeah? As I said already, Thomas Hillebrand, Malav Shah, Oliver Heise, Noemi, Tabea Potek, you should read, not Potek, yeah? So here, you see that Linda is one of the elephants we dissected. Yeah, here you see Thomas. Yeah, he's an elephant vet. He gives us Thomas Hillebrand. He gives us the elephants. Yeah, and what we did is we collected a lot of elephant brains. Yeah, this is different from other research. Yeah. Usually, most of the papers are on one elephant, yeah, N of one things. And with this N of one thing, you can see a lot of things, but you cannot get certain aspects that I want to talk now about. And I think it's very important to look at multiple brains. Yeah? Now, a lot of the brains are not in all that great shape. Yeah, so these come from zoo elephants. Yeah. Most of the brains, yeah, these die of old age, yeah, and then there's a problem. The elephant drops, yeah, and the zoos, they usually rely on the elephant walking around. Yeah, they don't really have means to move this 4-ton animal. Yeah, and then there's a big problem. The elephant dies, yeah, and then you come there the next morning and you touch him, and he's still warm. Yeah, they don't cool down, and all of this, it's bad for the brains. Yeah, so our brains. Not so great. We have now actually worked on this very hard, and we collected also some elephants that were electively killed in the field because they came into conflict with humans, and then we have very short post-mortem times. But most of the brains are not so great. I want to briefly talk about the methodologies. Yeah, so the methodologies for brain research, they are very dominated again by the model systems. Pretty much every week or every second week, there is a new paper on how to trace connections in a mouse brain with a new synaptic trick or what, and it's really very, very impressive what can be done in the model system. It's fantastic, yeah? What is the problem is when we started working with the elephants, most of our techniques, they were from the last century. They were 100 years old, yeah, and not great. It makes you feel old, yeah. It's obviously if you use techniques from the last century, you will not be able to compete, yeah. And I think this is a mistake by neuroscience that there are too few techniques developed for large brains, yeah. And we make a tactical decision then. We want to change this, and what we put on a lot of efforts is for X-ray techniques, and particularly for neuroanatomy. I think these are game changers. Yeah, and I want to elaborate briefly on this. So, for example, we developed X-ray dense neural tracer, yeah, ICT. Yeah. Here I want to show you some of our micro-CT work. Yeah. Here we have an elephant hemisphere. It's the entire hemisphere. When we look at the entire hemisphere with micro-CT, we get a resolution of maybe 70 voxels, 70 micrometer voxels. When we zoom in, we can now get down to 26 micrometer voxels. Yeah? Pretty good. And with this, you can see small things. For example, there's these little islands here. Yeah, it's a claustrum. Yeah? Interesting structure. When you look at this structure with the MR, you just don't see it because the resolution is not good enough. When you then get all these claustrum islands in the entire brain, you get a feeling for how the elephant claustrum is distributed. You find it's very distributed. Yeah, I don't know how many here are from the neuroscience community. There's a famous paper by Crick, yeah, Francis Crick from the DNA thing, that maybe the claustrum may be the center of convergence or so. I don't think this is how this looks. There's maybe 20% of the cells are in the main nucleus. Yeah, it's true. And there's a lot of convergence there, but most of it is very distributed. So looking at this across the brain, it reminds you more of a cortical layer than it would remind you of a singular convergence center. Yeah, the Crick had this idea that it might be the attention map or sort of something very high level, something like Descartes' little epiphysis theater. Yeah. Okay, cool. Let me talk more about the resolution improvements by X-rays. Yeah. When we started 4 years ago, we did mainly MR scans, yeah, and the resolution about 500 micrometer. Yeah, the zooming in abilities of the technique, they are quite modest. Yeah, I showed you this now, what we have with hemisphere. What we hope to do, or what we have imaging time this year in Grenoble, yeah, at the synchrotron. And the synchrotron is particle accelerators. You send the electrons around, and to keep them on course, to keep them in the circle, you need to deflect them, slow them down, and then send out X-rays. Yeah, and a lot of X-rays. Yeah, there is here a nuclear power plant. Yeah, so it's climate friendly, our research. Yeah, so. There, here at the BM18, they have a special beamline for large pieces, yeah? And they did incredible work on human brains and stuff. And we want to have an elephant there this year. So I hope that we can get brain widths to 10 micrometers or so. I think if we zoom in, it goes down to maybe 2 micrometers or so. This is a very interesting number for the cellular biologists, because then these large brain imaging techniques, they start to turn cellular. Yeah? And I think there's going to be a lot of change in large brain neuroscience from this ability to visualize at cellular resolution things in 3D. And yeah, that's where we put our money. And the experience from looking at more high-resolution things, if you have the images in 3D, it's an incredible advantage. The person with whom we do most of this, Tim Sal did, we collaborate heavily with Tim Salit, it's X-ray physicist, he calls it, it's dimensionality stupid. Yeah? It's somehow incredibly important to have it in 3D. Yeah? It's surprising, even if you do not have so much better resolution. When you have the images in 3D, you discover a lot that you just couldn't see this from the 2D images. So I think this is why this will get very important. Okay, let's look now at these brains. Yeah? There one thing quite interesting is sort of the development of the brain. This is a newborn male African elephant, adult male elephant, really big brain, yeah, 7 kilogram brain or 6.7 kilogram brain, yeah. You see immediately a very different size, yeah, a threefold increase, yeah. This is unusual in large brain animals. Yeah? In marsupials, you might have something like this, but in large brain animals like primates or so, this is very unusual. So the monkeys, if you plot how much brain they have at birth, yeah, they have about 60% of their brain weight at birth. Yeah? A couple of monkey species here. So they add 1/3 of brain as they grow up. Yeah? The apes, with the exception of the gorillas are like monkeys, yeah, but the apes, yeah, they add 60% of their brain as they grow up, yeah. The humans of primates, we add most, yeah. We add about 70% of our brain, yeah. We start with about 29% brain weight, baby, yeah, and then we add a lot. The elephants, they sort of fall in between the apes and the humans. We think this is related to this extremely experience-dependent behavior. Yeah? That's what people in the field say. They can even show when the old matriarch, the leading cow is not there. Somehow the group of elephants, they make less good decisions. Yeah? So you can really even demonstrate this in the field. Yeah? I would also say here, so the elephant's a bit like us, yeah? I think this comparison might be even a bit unfair to the elephants. And the reason is this very long gestation, yeah? So the elephants, they are born after 22 months of gestation. Very, very long gestation, yeah? And the baby is, in absolute terms, it's large, 100 kilogram, yeah? But in relative terms, it's small. It's smaller than the human baby, yeah, because the cow might be 3.5 tons, yeah, 100 kilogram, not so big, yeah. The human baby is bigger, yeah. So probably they delay the birth of their babies a long time to have them very advanced, yeah. Still, even though they are born very late, they add so much brain. Yeah, if you would compare the same time after conception, they would add a lot more brain than humans. I'm not sure I have explained this well, but let's move on. So a lot of experience-dependent growth. Yeah.
I had a small question about the graphic. Why is Homo sapien listed twice there?
2 studies. Yeah.
Of course.
They did 2 collectives of babies, and yeah. The reassuring thing, the result very similar. Yeah? So I think it was 2 cohorts of babies and adults. Yeah. Okay. I wanted to talk about another thing that I already alluded to, differences between Asian and African elephants. The Asian elephants, they are smaller. They are 10–15% smaller than the African elephants. Yeah? Turns out, however, the Asians have quite a bit bigger brain. Yeah? About 1 kilogram. Yeah? About 1 kilogram more brain, yeah, than the Africans. Yeah? I tend to think that this is very likely the most important difference between the 2 species. Why? Because the brain is so much more important than the ears. Yeah? There are very interesting differences between Asian and African elephants that we do not understand so well. One of them is that semi-domestication. Pretty much in every Asian country where there are elephants, you have semi-domesticated elephants, yeah? You have working elephants everywhere in Asia, yeah? In Africa, very rarely, yeah? It's not never. Also Hannibal, he had African elephants, but it's very rare to see sort of these African work elephants, yeah? And probably the reason is these behavioral differences, yeah? So the Africans, they are more impulsive, yeah, more fierce, yeah? If you annoy them, they push back immediately, yeah? And it's very different from the Asians, yeah? Much more deliberate, yeah? So if you cross an Asian elephant, he might do nothing, yeah? And then 3 years later, when there's a good opportunity, they kill the keeper, yeah? They operate quite differently, yeah? So I think this is a real thing and an interesting thing, yeah? Yeah?
Do you know anything about the brain size of zoo animals and wild animals? If it's experience dependent, could we expect?
I'd say the brain size is similar ad hoc, but we want to look now at brain parts. We are very interested in brain parts, and something we are looking at, in particular, we reason that the zoo elephants have a kind of restricted spatial experience. Yeah, the enclosure is probably smaller than this room for all their life. Yeah, scaled by body size. Yeah, the elephants in the field, hundreds of kilometers. Yeah. So we are very interested if there are differences, say in the hippocampi, and maybe I think we could probably figure this out. We are doing this, but I don't know the result. I know a result that I want to talk about now. And first, I didn't pay attention to this, but I think it's probably something very important. And that is, we sort of think that elephants are less equal than other animals. Huh? What do I mean by that? We stumbled across this when we looked at brains. Yeah? The brains, here we have two brains. Yeah? We have, I think, 8 or so female African brains. Yeah? It's the largest one, smallest one. Below 4 kilogram, more than 6 kilogram, 160% difference. That's an insane difference. Yeah? When you collect 100 human brains, yeah, you find such a difference. Yeah? But not when you collect 5 or 10. Yeah? So when you look at the coefficient of variation, segregating sexes and stuff, of brain weight in elephants, they have by far the most variable brain size in all mammals. Yeah? There are the elephants and all other mammals. They all have sort of a coefficient of variation of 8%. Yeah, the standard deviation is about 8% of the mean. Yeah, in humans, in monkeys, in carnivores. Yeah, what have you not? The elephants roughly twice that. Yeah, so very, very different brain sizes. Yeah, it turns out also other behaviors are very different from animal to animal. For example, something that's very easy to classify is sleep duration. Sleep duration's insanely different across elephants. So elephants have kind of the shortest sleep of mammals. Yeah, part of that might be that they need to feed so much. Yeah? They can get up early, and then if you want to get 18 hours feeding, there's obviously not so much time for sleep. Yeah, so insanely different sleep duration. So here in the zoo, we have one elephant sleeps an hour. There's another elephant sleeps 3.5 hours, almost 4. A fourfold difference, yeah? Much different from humans, yeah? In humans, there is kind of Napoleon, 3 hours, yeah? And then there's the rest of mankind, between 5.5 and 8 hours, yeah? We all sleep pretty similar lengths, yeah? It varies an hour more, but it never varies fourfold, yeah? There's nobody who regularly sleeps 22 hours, yeah? It's just not happening, yeah? So sleep duration. I told you already about grasping. The way they grasp things, there's this left and right trunk, very different. They're all the complex behaviors, I show you a few more complex behaviors. They are very different from elephant to elephant. Now, why is that so? Why are elephants so insanely different? So I think this relates to how they live. They are these 4-ton feeding machines, yeah? And there is just intense competition in the group, yeah? In the zoos, there are always fights when you feed the animals, yeah? Because they feel the pressure. For example, this banana peeling that I showed you. She would never peel a banana when she's with other elephants. It's only when you put her aside, yeah, then she peels it. But if there are other elephants around, she always eats it right away, yeah, because there's no time, yeah. You compete with these feeding machines, and sort of what we have, it's just a guess, obviously. We have this adaptive individuality hypothesis. We wonder if the elephants in these matrilineal groups, that they behave very different on purpose, yeah? That it reduces competition in this group. This is, of course, a post hoc. It's just a hand-waving explanation. But it's interesting that elephants are so much more different than other mammals. Let's look a little bit at what's inside these brains. The Asians have these bigger brains, but they have a lower cell density. Yeah, so they have a lower cell density. The Asians, they have maybe about 3,500 neurons or 3,000 neurons per cubic millimeter. The Africans maybe around 5,000 neurons per cubic millimeter. Those are very low numbers, very few neurons. Yeah? A mouse, it has 100,000 neurons per cubic millimeter. Yeah, 100,000. Yeah, humans maybe 25,000 neurons per cubic millimeter cortex. Yeah, the elephants, and this is something that holds true in general for large brains. There are just very few neurons. Yeah, very few neurons. And a thing that we don't understand why that is so. We have a hunch now, but it's a surprising observation. Interestingly, when you then calculate how many neurons are there in total in the brain, so we would get sort of the volume, the gray matter volume for different elephants, yeah, from MR images. We would determine the density, and then calculate a total number of cortical neurons. It's maybe about 4 billion in Asians and 4 to 5 or so in Africans, not significantly different. Even though the Asians have a kilogram more brain, they don't have more neurons. And what we think then is these larger brains, they are not made larger for more neurons. Yeah? This is something that's pretty clear to us. Yeah? What is it good for then? So we do a lot of high resolution work, yeah, synchrotron, where we look at these networks, yeah. And here we compare the elephant and the smallest mammal, yeah, the Etruscan shrew that we also work with, yeah. A 5 kilogram brain, a 60 milligram brain, the smallest mammalian brain, yeah? When you look inside, what do you see? This is the elephant, that's the shrew, yeah? You see these big neurons, yeah? And you see so many here, yeah? A big number difference, yeah? Quite striking, yeah? When you zoom in, one thing that is clear to us that is more in there is the cables. They are just much thicker, yeah? And it was already known that the dendrites, these are these big neurons, yeah? This is an elephant neuron, yeah? This is a shrew neuron, yeah? Look at the size difference. So the dendrites, much thicker, yeah? And here these black things, they are myelinated axons. Very impressive in the elephant. Yeah, in the shrew you don't even see that. If you would take a mouse brain in between, a mouse is a 500 milligram brain, maybe 10 times bigger than this. Yeah, it would look like in between. You would see some myelinated axons, but they would be very small. They would be like these or so, just visible. Yeah. So what we think something that is positively in there is very thick cables, very thick dendrites, and very thick axons. Yeah. So what we tend to think that these large brains are maybe built for speed rather than for more neurons. Yeah? And I told you this thing, that you sort of need to slow down the movie to really see what the elephant is doing. And we think these very fast behaviors and these very big cables, yeah, that this might be related. It's just a guess. Yeah?
I would like to understand better. I just thought that because the signal has to travel such a long distance in the brain, you need the more myelinization.
Yeah.
You have more or less the same time for signal process. So at the end, does this mean that the signal still travels faster in the
I would bet.
The distance that it has.
Yeah, I would bet the signal's going to be a lot faster, because this is something clear to us. The cables are so much thicker and there's so much more myelization and stuff. Yeah, how much more exactly I cannot tell you, but we'll find out. A few months. Yeah, we try to do it very quantitatively at the moment. Yeah, but a big difference. It's a big difference between small and large brains. So does it compensate for the size? I don't know. But I think sort of high resolution, even higher resolution images of these brains, that will be very helpful. And then being quantitative about these differences. I think maybe they just look larger, but factually, because the cables are so much faster, maybe the distances between the neurons are not even that much bigger. But it's just all fantasizing. Yeah, we want to get quantitative numbers on that. Yeah.
Thank you.
I skip this because I'm going over time. You see also the wrinkles in the brain, but I want to talk now a bit about house tool use. Work by Lea Urban, Lena Kaufmann, Rolf Becker, and Andreas Ochs in the zoo. It was discovered by Jane Goodall that chimps use tools. Turns out, actually, many animals use tools. Yeah, quite a few. Yeah. What is the case, however, most of the animals' tools are pretty simple. Yeah? It's pretty simple tools. Yeah? Here we look at houses. Yeah? Houses are a bit like the trunk. Yeah? The trunk, if you want to think about it, is like a house. And we wonder if that's why elephants are so good with houses. Yeah? They're so fun. So what is interesting about houses are very extended. Yeah? The experiments we do, the houses, they are bigger than the elephant. Yeah, and that turns out to be important in these experiments. Yeah, also houses are complicated. Yeah, you have the water flow. A house sort of functions in a holistic manner. Sort of when there's a leak in a house, it's useless. Yeah, I teach a patching course. There's always a leak in a house. You can't use the setup. Yeah. Okay. Importantly, for these experiments that I talk about. The elephants, they are trained not to step on houses in the zoo. Yeah, they are not allowed to step on houses, and you would actually you see them holding the hose with a paw, but they would not step on it. If they step on it, the caretaker yells at them. Yeah, because it slows down the showering and all that. Yeah, so they are not allowed to do that. Yeah, that's important to understand that. Here, let's look at Mary. She's kind of very good with houses, yeah, and she's very good in showering. There is no other elephant in the zoo that showers like her. Yeah? It's one of these many things where the elephants. Now she takes a drink, yeah, and. Then she showers again. And we are very impressed how well she has understood hosts use. Yeah, so she gets behind spots that are difficult to reach. Yeah, here behind the ear, and in a second you will see her showing her legs. So there's a lot of body tool use, coordination. Yeah, so she brings up the hind leg. Yeah, and then showers it. It's really beautiful. Yeah. She very much uses the hose like a shower head. Yeah, so she takes it up. Now she uses the forelimb. You see that? That's a typical Asian elephant behavior. African elephant would never do that. And she holds it sort of 20 centimeters from the tip. Yeah, that's how she holds it. 90%. There's an interesting exception to that. When she wants to get to her back, she uses a different strategy. Yeah. Now she grabs it sort of a meter behind. Yeah. And then she slams this thing on her back. Yeah, she kind of uses the ballistics of the hose. Yeah, to get to her back. Yeah, and amazingly different uses of the tool. Yeah, you also see it looks very joyful. Yeah, that's obvious. When you ask the caretakers what they think about this, they would say, yeah, it's very joyful, but they would also say it's also a bit posing, yeah, sort of. She has a hose, so it's like, look at me, I shower, I have the hose. Yeah? I don't see that, yeah, but then you talk to them, and when you then know it, you get the feeling, yeah. And guess what? This doesn't sit well with the other elephants. Yeah? So the other elephants are not pleased. And it's kind of the context of this research project. Yeah? We went there. She gets a lot of shower time. Yeah? There is a lot of envy. Yeah? And then we saw this behavior, which I think was one of the most stunning behaviors that we saw. Yeah? And it's again Mary showering here. Yeah? But then there's Anjali. Yeah? And I thought she wants to steal it, yeah, when I saw this. But it's not what Anjali is doing, yeah? So actually what Anjali does is she makes a kink into this thing, and then she squeezes this kink like hell. And basically what we think she's trying to do is she's trying to stop water flow to Mary, yeah? And if you look at it in slow motion, you see that. So first she lifts the hose. It has absolutely no impact on the water flow. Yeah? Then she kinks it. It also doesn't affect the water flow. It's very hard. It's very, very difficult to stop the water flow through these big hoses. Yeah? Then she re-grasps this kink. And also that it still doesn't stop the water flow. But then in a few seconds, now watch the water flow here. In a few seconds, she starts squeezing like hell, and she will lose the hose in just a second, yeah? But now look at her. It's still flowing, and now she stops it, yeah? Oops. And then she immediately loses it, because she needs to squeeze like hell to make the water stop, yeah? And over a couple of days, she perfected this behavior, so she got actually much better in stopping the water flow to Mary. And what we think is happening there is sabotage. Yeah, she's trying. It's not about having the hose or so. It's a behavior about the hose. Yeah, and why do we think so? I can't go into all of that, so let's show maybe here this briefly. So this is, she first positions the hose. The first time we saw the behavior, she positioned the hose most carefully for 18 seconds, yeah, very, very carefully. The longest time that we ever saw this, yeah? And we think at that time she thought about this, yeah? And then she did the things, she lifts it, she kinks it, she regrasps the kink, and then she contracts, yeah? And then she stops the water flow. The black here is the closure, yeah? And over a couple of days, she got better at it, yeah? Sort of, she managed to stop the water flow about twice as long, yeah? So an amazing behavior. It's really a behavior more from a comic than from a zoo. Yeah, these sabotage things, you don't usually see that in animals. There are some orcas of the Spanish coast. They seem to purposefully incapacitate sailing boats. Yeah, they take the rudders away. Yeah, it looks like sabotage, but it's very rare. Yeah, most interestingly, elephants. There are other sabotage behaviors described. Yeah. I conclude elephant brains and trunks. Yeah, brains very neglected. Yeah, the Asians have larger brains than the Africans. There's this massive postnatal growth. We wonder if these large brains are more about speed, yeah, thick cables, than about adding neurons. Yeah, many people they counted the neurons. Oh, there are not so many neurons. That's why they are no good. I think I doubt this very much. Yeah. When you look at trunk behavior, at elephant behaviors, they really do qualitatively different behaviors than, say, rats and mice. Yeah, and if you look at them doing things, it very much makes you wonder if their intelligence doesn't have something to do with these manipulative skills from the trunk. Yeah, these very extraordinary behaviors that you just saw of Anjali. So the sabotaging, I think this is related to these manipulative abilities. And if you look across animals, I think there is reason to think that this is probably a good idea. This idea, I think I read from it first from Konrad Lorenz. Yeah, he had this idea that the grasping hand is very important in the evolution of human intelligence. Yeah. If you look at octopuses, you know, very extraordinary, you know, little mollusks. Yeah, you wouldn't expect them to be smart, but they can also do things with their arms. Yeah, so I think in intelligence, this manipulation might be very important. And here again, I want to advertise this more across scales approach. You need to look into the field. You need to look into the whole brains. I think you need to look also at the nanometer level. Yeah, at how the cables are, if you really want to understand animals. And that's it. Thank you for your attention.